there's going to be a lot of the same things we'll see in this that were in the production of an immunoglobulin molecule. Does everybody have this handout? Okay. So, again, here's our Y-shaped molecule that we call an immunoglobulin. The T-cell receptor, and the thing is there are two versions of it. We're going to focus on the alpha-beta, but I'll tell you about the other version as well. But if you look at this T-cell receptor on the right side, it looks a lot like just an FAB fragment from an antibody. Just one of the arms, and then it's got an alpha chain and alpha, just these two chains that form an antigen binding site up at the top. And so we'll have this alpha and a beta chain. We have the variable region, we have a constant region, we have a transmembrane region, and very, very little cytoplasmic tip. Because, and there's no way for this thing to signal by itself. We'll see how it acquires the CD3 that we talked about earlier that plays a role in signaling through this molecule. So these are, in very general terms, the phases that we're going to talk about, phases of T-cell development. This is where the T-cell differs from the B-cell. The T-cell progenitor and the B-cell progenitor both are made through hematopoiesis in the bone marrow. At some state, the T-cell gets the wild idea that he wants to be a T-cell, and he leaves the bone marrow, goes to the thymus. And this T-cell is a very generic thing. It doesn't look anything like a T-cell. It doesn't have the markers on it like a T-cell. And so it moves to the thymus to try to see if it's going to eventually become a T-cell. So immature T-cell goes into the thymus, and it goes through the process of rearranging the genes that are involved in this. That's an alpha chain gene and a beta chain gene. And what we're going to find is that they're analogous to a heavy chain and a light chain. One rearranges first, and then the second one does. One has V, D, and J, and one just has V and J. But they rearrange these genes. If they develop a functional T-cell receptor, then they test it. They will go through positive and negative selection to make sure that they work, that the T-cell receptor works, and that it doesn't work too well. Does it recognize self-antigen in the context of self-MHC? If it does very strongly, that cell's got to die. You've got to get rid of it, because that will cause an autoimmune disease bigger than anything you've ever seen. After they've gone through positive and negative selection, they have acquired the phenotype that we know. CD4 if they recognize class 2, CD8 if they recognize class 1. This is MHC restriction. That is what this means right here, is that these T cells are restricted to what MHC they recognize based on whether they're CD4 positive or CD8 positive cells. So you're going to hear CD4 positive cells a lot in the future, I'm sure, because that's one of those things that we talk about in immune status of patients. If CD4 numbers get too low, that's an immunodeficient state. And that's what happens in AIDS. HIV actually binds to the CD4 molecule, gets inside these T cells, and kills the CD4 T cells. And remember I told them, if the immune system was an orchestra, the CD4 T helper cell is the conductor. You knock out the conductor, then you've knocked out the ability to really respond well to a lot of infectious diseases. People are not dying from AIDS, they're dying from infections following AIDS. Pneumocystis carinii comes in, you have some Kaposi sarcoma, you have some other diseases that typically your body can handle when you have functional CD4 T cells, and if you lose them, you don't. All right, latest panel there, we've talked about multiple times. The T cell goes to the secondary lymphoid tissues. It interacts with the antigen-presenting cells there, most likely dendritic cells. They become activated if they run into their antigen. They leave and go look for their antigen in the periphery, and if they find it, then they do their thing at that place. If they're a cytotoxic CD8 positive T cell, they kill. If they're a CD4 positive T cell, they help in a number of ways, which I will get into a little bit later. And so we'll go through these phases in a little more detail. Here's the low side. You got to have some genetics, because we did it with the B cells. And these are what the two loci that we're going to be most interested in look like. You have on the top the alpha chain, and you see it has a V group, 70 to 80 V alpha gene segments. And again, what's upstream of them? A promoter. Each one of them has their own promoter. That red box, 70 or 80 of them, whatever they are, there's a promoter upstream of each one of those. Downstream of that are the J segments, and this particular one showing you 61 different J segments. So you have approximately 80 V alpha that can interact with approximately 61 J alpha gene segments to produce the variable part of the alpha chain. And then you just have one constant region gene. You only need one in the T cells because they don't have an effector function like the B cells. If we go down to the bottom, we've got the beta. And then the germline, we have 52 of the V gene segments. And then we have two groupings based on D and J. And so we have D beta 1. There's one of those associated with J beta 1, six J beta 1s, and then we have the D beta 2. And so those are going to interact together. And then you have the constant region part just below it, just past it, downstream of it. And so what we're going to find is that, like with the beta chain, or like with the immunoglobulin, the loci that has the D segments is going to start rearranging first. D to J is going to be a first rearrangement, and then V to DJ as we go through it. And we're not going to go through this in great detail, but this is something, I showed you another picture that had the ITAMs on it a long time ago, first thing we looked at. The alpha and beta T cell, the alpha chain and the beta chain of the alpha beta T cell receptor do not have the signaling motifs that are required to send a signal to the cell. So before that can go up onto the surface of the T cell, it has to associate with these other things that we call CD3. And there's all these, well, there's six proteins, but some of them are identical. There are 2 epsilon, 1 gamma, 1 delta, and 2 zeta that are associated. And all of them have those immunoreceptor tyrosine activation motifs associated with them, ITAMs. You also notice that they have charges associated with them. The transmembrane region of the alpha-beta T cell receptor has charges, opposite charges, and so these things do associate with each other in the membrane for signal transduction. And again, like we saw with immunoglobulin, remember we had a heavy chain with 3 CDRs and a light chain with 3 CDRs that come together, and the 6 CDRs are the antigen binding pocket. We have the same thing in the T cell receptor. The alpha chain has 3 CDRs, the beta chain has 3 CDRs, and so the 6 together form the antigen binding part. But in this case, it's not recognizing something globular, it's recognizing MHC molecule with a peptide in the middle of it. All right, what's next? Now, I mentioned that there were 2 types of receptors. And we'll just talk about this for a minute and take a little break. The alpha-beta T cell receptors are the ones that we have studied the most. That's your CD8 positive T cells, CD4 positive T cells. They're going to have the alpha-beta. There's this other group called gamma delta T cells. And they're called that just because, well, alpha and beta is taken, so let's go to the next Greek letters, gamma and delta. And these are a little bit different in that they do probably recognize MHC molecules, but they also recognize some non-classical MHC molecules, and they don't have a CD4 or a CD8 type molecule associated with them. But when a T cell is, when a cell is trying to go through the process of becoming a T cell, it has to go through the process of deciding which one it's going to be. It can't be both. You can't have both of them on the cell surface, or you'll have multiple specificities. But there are mechanisms to make sure that we exclude one of them from the surface if we've chosen one of the others. And we'll look into that as we go through this. This we're going to get at how we kind of make the T cell receptors as we go through this. You've already seen this particular slide, but we're going to start putting these things together. We're going to start rearranging the beta chain levers on the bottom first. And I think what we'll do is we'll stop right now, come back in 10 minutes, and pick up there and move forward from that point, talking about gene rearrangement and T cells.